So this was a phase 1 dose escalation study of PHE885, which is a fully humanized anti-BCMA CAR-T cell product for patients with relapsed refractory multiple myeloma that's produced using this novel T-Charge platform, so it allows manufacturing in under 48 hours. This was a phase 1 dose escalation study looking at PHE885 in patients with relapsed and refractory multiple myeloma who have been exposed to at least 2 prior lines of therapy and had been exposed to an IMiD, a PI, and an anti-CD38 antibody. So it's an autologous CAR-T product targeting BCMA, and we were able to manufacture the product using this novel approach that allows manufacturing to occur in under 2 days that will improve the return of product to patients. So we'll shorten the manufacturing period and allow patients to be treated faster. And so we tested multiple cell doses ranging from 2.5 to 20 million cells in a total of 50 patients. And we were able to see a few things. The first is that because this manufacturing approach is much shorter, the cells are ex vivo, or are sort of being grown ex vivo for a much shorter period of time. They retain some of the sort of more stem-like or memory-like properties within the T cells. So we think they're a little bit healthier and they may have better function, persistence, and expansion. We saw very good expansion in patients, so we can infuse as few as 2.5 million cells and see the cells expand within patients. And we saw a very, very high response rate. So 98% of patients responded. So there were 50 patients on study, so only 1 patient did not respond to this treatment. I think we've made huge advances in the last few years in terms of developing CAR-T cell products for patients with relapsed and refractory myeloma. But one of the problems is that those cells take a fair amount of time to manufacture, often 6 to 8 weeks to return the product. And there are many patients who have aggressive disease who just can't wait that period of time. So I think developing a manufacturing process that is faster will be able to get these really effective therapies to patients who couldn't otherwise get them. And I think another big problem is that a lot of patients who receive CAR-T cells, they still relapse. And so we need better products. And so the goal here also is by making a T-cell product that maybe is a little bit healthier, that has more of a memory phenotype, maybe it'll function better. I think we don't have the follow-up to know whether or not that's the case, but that's one of the hopes from this study as well.